discussing factors affecting. We were discussing the factors affecting the enzyme. Affecting the enzyme activity. Factors affecting the enzyme activity. So under factors affecting the enzyme activity, what are the topics we have discussed? First was pH. Then what? Temperature.
Yes, ma'am.
Then
Concentration of substrate.
Substrate. And after concentration of substrate, next factor was cofactor.
Yes, ma'am.
Yes, ma'am.
And the last thing we were discussing is inhibitor. Today under the inhibitor only, we will discuss the 3 types of inhibitor. Okay? So we were discussing all these factors, and out of that, the last class, the last topic was inhibitor. So from the name itself, inhibitor will reduce the enzyme activity. Okay? There are 3 types of inhibitor: competitive inhibitor, non-competitive inhibitor, and allosteric inhibitor. So we will discuss in details all 3 of them. Okay? So there are 3 types of inhibitor. Competitive inhibitor, non-competitive inhibitor, and allosteric inhibitor. So in today's class, we'll discuss in details about 3 of the different types. See, competitive inhibitor. From the name itself, competitive. There will be someone for competition. Okay, but who will compete? We know that enzyme has a site known as active site. Okay, where the substrate will bind. Okay, every enzyme have a specific active site where a specific enzyme can, or specific substrate can only bind. Eta enzyme or যেটো এনজাইম অ্যাক্টিভ সাইট হয় তাতে কি হ'ব পাৰে অকল এটা স্পেসিফিক সাবস্ট্রেটে বাইন্ড কৰিব পাৰে বেলেগ সাবস্ট্রেটে বাইন্ড কৰিব নোৱাৰে ওকে নহলে ৰিঅ্যাকশনটো কেনেকৈ হ'ব যদি সব সাবস্ট্রেটে বাইন্ড কৰিব পাৰিলে হয় তেতিয়াটো কথাই নাছিল এটা এনজাইমৰ স্পেসিফিক সাবস্ট্রেট এনজাইম বিলাক কি হয় সাবস্ট্রেট স্পেসিফিক হয় ওকে তো অকল পার্টিকুলার সাবস্ট্রেটটোহে আহি পিনে কি কৰিব পাৰে ইট ক্যান বাইন্ড টু দা এনজাইম but what happened in case of competitive inhibitor what will happen another substance which have same structure like the substrate will come and bind to the enzyme active site. Another substance which is having the same structure like that of the substrate, original substrate, will come and bind to the active site of the enzyme. As a result, what will happen? The active site is blocked. So now the substrate cannot bind to the enzyme. As a result, product form hobo ne nahoy. Product form hobo ne nahoy. Product form hobo. Substrate যেতিয়া enzyme bind কৰে তেতিয়া enzyme substrate complex হয় which further gets converted to enzyme product complex. যদি substrate enzyme bind কৰা নাই তেতিয়া product form hobo. Question mark?
No.
No, okay?
No, ma'am.
So remember, competitive inhibitor ki koribo substrate or jagatu loi lobo. so that enzyme substrate to bind koribo nore aru that competitive inhibitor will bind to the enzyme. As a result, the reaction is not proceeded. Okay, reaction to rokhi jay. So let us see what will happen in case of competitive inhibitor. A substance which has similar structure like that of a substrate like that of a substrate of a particular enzyme binds to the active site of the enzyme. As a result, the substrate cannot bind to the enzyme. As a result, the substrate cannot bind to the enzyme, so no product formation takes place. No product formation takes place. So no product formation takes place. A substance, in case of competitive inhibitor, a substance which has similar structure like that of a substrate of a particular enzyme binds to the active site of the enzyme. As a result, the substrate cannot bind to the enzyme, bind to the active site of the enzyme. So no product formation takes place. So no product formation takes place. Have you written?
Yes, ma'am.
No, ma'am, that's what you.
Okay, okay. Write it down.
Yes, ma'am, done.
Okay. So here the substance is competing with the substrate. Substance to কাৰ লগত competition কৰিছে? Substrate লগত competition কৰিছে, substrate জাগাটো লৈ লৈছে। যদি competitor টো নাথাকিল হয়, যদি এই substance টো নাথাকিল হয়, তেতিয়া হলে কি হল হয়? Substrate would be binding to the enzyme. But এতিয়া কি হৈছে? That substance is a competitor to the substrate. So this type of inhibition is known as competitive inhibition. Okay. So write one more line. The substance is competing with the substrate for the active site of the enzyme. So the substance is called a competitor and the type of inhibition is called competitive inhibition. And the type of inhibition is called competitive inhibition. And the type of inhibition is called competitive inhibition. The substance is competing with the substrate or active site of the enzyme. So the substance is called competitor and the inhibition is called competitive inhibition. Done.
Yes,
Now let us
ma'am.
come to the example. The example is given in your NCERT book. Succinate is the substrate which reacts with the enzyme succinate dehydrogenase to give fumarate. Okay, so succinate, suppose this is the substrate, succinate is our substrate. It reacts with the enzyme succinate dehydrogenase to give fumarate, which is the product. So our enzyme is this is our enzyme. This is our substrate. This substrate should go and bind to the active site. But there is a competitor. The competitor is malonic acid. So this is the competitor, malonic acid. This is our succinate And this is our competitor, malonic acid. This malonic acid will prevent, this malonic acid will go and bind to the active site. Okay. As a result, the substrate cannot bind. Okay, it cannot bind. So as a result, no product. Okay. Got it? বুজি পাইছা? কি হয় সাবস্ট্ৰেটৰ কাম হল যাই পিনে এনজাইমৰ লগত বাইন্ড হোৱা, এনজাইমৰ এক্টিভ সাইটত বাইন্ড হোৱা। বাট সাবস্ট্ৰেটৰ নিচিনা সিমিলাৰ দেখা, সিমিলাৰ স্ট্ৰাকচাৰ থকা এটা কম্পিটিটৰ আহিব, সেই কম্পিটিটৰটো আহি পিনে কি কৰে, ইট উইল গো এণ্ড বাইন্ড টু দ্য এক্টিভ সাইট অফ দ্য এনজাইম। এজ এ ৰিজাল্ট কি হয়, সাবস্ট্ৰেটটোৱে এনজাইমক বাইন্ড কৰিব নোৱাৰে। সো সাবস্ট্ৰেটে যদি এনজাইমক বাইন্ড কৰিব নোৱাৰে, সো প্ৰডাক্টটো ফৰ্ম হ'ব নোৱাৰে। ক্লিয়াৰ এভৰিৱান? সো নোট দিস ডাউন। So succinate, you write, this is the substrate and malonic acid is the competitor. Malonic acid is the competitor. So malonic acid will take the place of the substrate, that is the succinate. Done?
Yes, ma'am.
Yes, ma'am.
Okay. So you can add one point. In the above reaction, malonic acid acts as a competitor and binds to the active site of succinate dehydrogenase binds to the active site of succinate dehydrogenase, thus preventing the binding of succinate after that you can continue here to succinate dehydrogenase. As a result, fumarate, product fumarate is not formed. As a result, product fumarate is not formed. Right, this is example 1. There is another example which I will give. Have you completed?
Yes, ma'am.
So next example is example 2. You write Papa. Papa's full form is in presence of folic acid synthesis. Synthesis. Let me check the spelling. Synthetase. Para amino benzoic acid. Okay. Para amino benzoic acid in presence of folic acid synthetase produce folic acid, which is required for the growth and development of bacteria. Which is required for the growth and development of the bacteria. So here, in this reaction, what will happen? Sulfur drugs. There are some drugs known as sulfur drugs. It acts as a competitive inhibitor of GABA. Okay. In the above reaction, sulfur drugs act as competitive inhibitor of whom? Car competitive inhibitor of. What is the substrate here?
Paba.
GABA. Okay. Competitive inhibitor of GABA. Para-amino benzoic acid. So the first reaction is given in your NCERT book. And the second reaction is although not given in your NCERT book, but Agote, when you will check the previous year papers, you will find questions based on this concept. Okay.
Ma'am, can you explain this example again? I got disconnected.
Okay. Like similar to the first example, here the substrate is PABA. PABA means para-amino benzoic acid. This para-amino benzoic acid will bind to the enzyme folic acid synthesis. Okay, in order to form folic acid. Okay, but here the competitive inhibitor, that is sulfur drugs, is the competitive inhibitor. It will come and bind to the folic acid synthesis. As a result, PABA will not bind to the enzyme, and as a result, folic acid won't be produced. Got it? So this is the substrate. This is the enzyme, and this is the product. Same concept. Last example, जितो concept असेल कि substrate और जगह तो competitor লৈ লয় আৰু as a result substrate enzyme লগত bind কৰিব নোৱাৰে আৰু product form নহয় same thing ইয়াতে substrate হল papa enzyme হল folic acid synthase আৰু product হল folic acid okay and this
Yes, ma'am.
reaction takes place in the bacteria because this is important for the growth and development of bacteria.
Okay, ma'am.
Next, write it down. One more point is there for competitive inhibition. Done, everyone? One last point for competitive inhibitor.
Yes, ma'am.
Yes, ma'am.
Competitive inhibition is a reversible process. How can we reverse it? By adding more substrate. If we add more substrate, then we can reverse the process. Okay? Very important point. Competitive inhibition is a reversible process the reaction can be altered, the reaction can be reversed by adding the reaction can be reversed by adding or by increasing the concentration of substrate by increasing the concentration of the substrate. So this is all about competitive inhibitor. Next, we'll discuss about non-competitive inhibitor. Done, everyone?
Yes, ma'am.
Next, we'll discuss non-competitive inhibitor. So, competitive inhibitor, the inhibitor is having same structure like that of enzyme because enzyme or যেটো active site structure হয় সেইটো specific substrate structure লগত same হ'ব নহলে তো bind হ'ব নোৱাৰে suppose মোৰ enzyme যদি active site টো এনেকুৱা shape হয় যদি মোৰ substrate টো circular হয় bind হ'ব পাৰিব active site লগত
No, ma'am.
No, ma'am.
okay so competitive inhibitor কি হয় substrate টোৰ যেনেকুৱা shape থাকে substrate টোৰ যেনেকুৱা structure থাকে same structure কাৰ থাকে competitor থাকে in case of non competitive inhibitor the structure is not having the same the competitor is not having the same structure like that of substrate so logically এতিয়া substrate to এতিয়া কি হ'ব the competitor will not bind to the active site it will bind to some other site but binding of the competitor to some other site will change the structure of the active site of the enzyme okay এটা কি হ'ব the competitor টোৱে কি কৰিব it is not structurally similar to the substrate so substrate or active site bind it can bind to the enzyme's active site এটা কি হ'ব it will bind to some other site present in the enzyme যেতিয়া competitor enzyme বেলেগ এটা সাইটত বাইন্ড কৰিব তেতিয়া enzyme কি হ'ব enzyme structure টো changes আহিব enzyme structure যেতিয়া change আহিব that means enzyme active site টো change হৈ যাব এতিয়া although substrate টোৱে active site bind কৰিব but এতিয়া product form still নহব got it মই কি কৈছো যে suppose এইটো মোৰ substrate হয় and this is my enzyme this time my competitor is not having the same structure মোৰ competitor টো এনেকুৱা হয় this is my competitor competitor যোনে কি কৰিব inhibit কৰিব so নামটো হ'ব inhibitor okay so ইয়াতে inhibitor লিখিবা better because ইয়াতে compete কৰা নাই হা inhibitor so inhibitor is different in structure so this inhibitor will bind to suppose this site etu site bind কৰিছে as a result কি হ'ব enzyme structure to change হৈ যাব okay এতিয়া যদি substrate টো bind কৰে substrate to suppose bind কৰিলে but still this is my substrate but still no product this is my inhibitor binding to enzyme in some other site okay আৰু ইয়াতে লিখিবা binding এইটো ডায়াগ্ৰামৰ তলত লিখিবা binding of inhibitor to the enzyme modifies the structure of enzyme modifies the structure of enzyme so substrate although binds to the enzyme cannot border এইটো part visible হৈ আছে ন
मैम completely নহয় অলপ অলপ
নাই completely হোৱা নাই
okay wait okay তোমালোকে diagram টো draw কৰা এইটো লাইন মই dictate কৰি দিম okay বুজি পাইছা substrate থাকিব substrate আৰু inhibitor structure এবাৰ different হয় বাট ইনহিবিটৰে এবাৰ এক্টিভ সাইটত বাইন্ড নকৰে, ইনহিবিটৰে বেলেগ এটা সাইটত বাইন্ড কৰিব, বাট ইনহিবিটৰে যদি বাইন্ড কৰে তেতিয়া কি হ'ব, এনজাইমৰ ষ্ট্ৰাকচাৰটো চেঞ্জ হৈ যাব। এনজাইমৰ ষ্ট্ৰাকচাৰটো চেঞ্জ হোৱাৰ লগে লগে অলদো সাবষ্ট্ৰেটে বাইন্ড কৰিব, ষ্টিল প্ৰডাক্ট ফৰ্ম নহয়। ক্লিয়াৰ এভৰিৱান?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Diagram, I will dictate that sentence, okay? আচ্ছা, এইটো বেচা সবে অসমীয়া বুজি পোৱাটো? Anyone who don't understand Assamese? So the line is, binding of inhibitor to the enzyme modifies the structure of the enzyme, so the substrate, although binds to the enzyme, cannot produce product. Binding of inhibitor to the enzyme modifies the structure of the enzyme, so the substrate, although binds to the enzyme, cannot produce product. Holger, have you drawn this?
Yes, ma'am.
Okay. See, now what can we write for the write-up? In non-competitive inhibition, the inhibitor is structurally different from the substrate. And the inhibitor does not binds to some other site of the enzyme, leaving the active site. In non-competitive inhibition, the inhibitor is structurally different from the substrate and the inhibitor binds to some other site of the enzyme, leaving the active site. Done. Next
Last round
graph
पे
you can.
देखो।
Good.
Yes, ma'am.
Okay. Next same line of continue koriba comma di pine but binding of the inhibitor changes the structure of enzyme. Therefore, substrate may bind to the substrate may bind to the enzyme, but product formation is prohibited. Non-competitive inhibition is an irreversible process. Okay, K2SL reversible process. Competitive inhibition was a reversible process, but non-competitive inhibition is an irreversible process. Next point, non-competitive inhibition is a irreversible process. Non-competitive inhibition is an irreversible process. Have you completed?
Yes, ma'am.
Okay. The next example is cyanide inhibits cytochrome C, okay, which is the substrate. Example for non-competitive inhibition is cyanide, which is the inhibitor of cytochrome C. Cyanide inhibits cytochrome C. inhibitor and this is the substrate. One more inhibition is there. Inhibitor is there. Okay, done. Next type of inhibition is allosteric modulation or feedback inhibition. Allosteric, third type. Okay, allosteric modulation or feedback inhibition. See, this type of inhibition occurs in chain of reactions. যদি এটা সাইকেলতে বহুত কেটা reactions একেলগে হৈ আছে তো এটা reactioner product next step to কি কৰিব পাৰে it can act as a inhibitor. Okay, so what you can write, glucose and ATP in presence of enzyme hexokinase. Glucose and ATP যদি আমি react করো in presence of enzyme hexokinase, it will give glucose 6-phosphate. Glucose 6-phosphate and ATP. This glucose 6-phosphate act as an inhibitor. Inhibitor of glucose. Keneke kore? I am explaining. Write down this reaction once. So, if more reactionat glucose 6-phosphate if I have 110 glucose 6-phosphate, but I need 100 glucose 6-phosphate. So, কি কৰিব? এতিয়া glucose 6-phosphate it will signal hexokinase. It will signal hexokinase negative signal দিব. So that production of glucose কি হৈ যায়? কমি যায়. Got it? যেতিয়া glucose 6-phosphate বেছি হৈ যায়. Okay, তেতিয়া কি কৰিব? It will signal hexokinase. It will signal hexokinase to reduce its work. Okay, so that glucose আৰু কি নহয়? Degrade নহয়. Glucose ওপৰত আৰু এক নহয়. Okay, so এইটো কি বুলি কয়? Negative inhibition. Positive দিব পাৰে, but আমি ইয়াতে negative টো চাই আছো. যদি suppose glucose 6-phosphate আমাক বেছি লাগে, suppose 120 লাগে আৰু ওলাই আছে 110, তো তেতিয়া কি কৰিব? Positive feedback দিব. But ইয়াতে আমি কোনটো context ত চাইছো? We are looking in the negative feedback. Okay, so it can stop the reaction. Okay, আৰু এইটো চোৱা glucose 6-phosphate actually product হয়. এটা reactioner product কি কৰি আছে? ওপৰৰ reaction to কি কৰি আছে? Inhibit কৰি আছে. মানে glucose of production to glucose 6-phosphate production to কমাব কাৰণে it is giving a negative feedback. Are you getting my point? Yes or no?
Ma'am, I will explain what you
Okay.
Okay. I'm explaining. Have you written? লিখি লোৱা. It's a
Yes.
explain কৰি আছো.
Yes, ma'am.
মোৰ তাত গ্লুকোজ 6 ফসফেট 110 টা আছে ওকে মোক লাগে 100 টা গ্লুকোজ 6 ফসফেট গ্লুকোজ 6 ফসফেট কৰ পৰা বনি আছে গ্লুকোজৰ পৰা বনি আছে isn't it গ্লুকোজৰ পৰা বনি আছে তো কি কৰিব গ্লুকোজ 6 ফসফেট যেতিয়া এক্সেস হৈ যায় তেতিয়া glucose 6 phosphate will give negative signal কাক দিব hexokinase দিব so that glucose কি হয় no more glucose is used ইয়াতে negative signal দিব no more glucose is used যেতিয়া glucose আৰু use নহয় তেতিয়া হলে কি হ'ব automatically কি হ'ব কমি যাব বুজি পাইছা এটা automatically কমি যাব না production টো got it মানে glucose 6 phosphate কি কৰিব পাৰে it can inhibit the reaction কেনেকে inhibit কৰিব পাৰে negative signal দি পিনে inhibit কৰিব পাৰে glucose ৰ production টো কি কৰিব পাৰে ৰখাই দিব পাৰে got it তো সেই কাৰণে এটো আমি কি বুলি কও negative feedback clear হৈছে
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
okay so it actually takes place negative inhibition feedback inhibition কেতিয়া হয় যেতিয়া chain of reactions হৈ থাকে the product কি কৰিব পাৰে নিজৰ production টো regulate কৰিব পাৰে so for the write up part what we can write so a negative feedback or question NCERT bookot nai so negative feedback or question very rare still keteba aile I am giving you this concept okay so for write up you write the activities of some enzyme the activities of some enzyme particularly that form part of chain reaction activities of some enzyme particularly that form the part of chain reactions are regulated internally so enzyme or activity কোনে regulate কৰি আছিল আমাৰ example টো কোনে regulate কৰি আছিল আমি যেটো example পঢ়িলো তাতে
hexokinase
hexokinase is the enzyme so hexokinase or activity কোনে regulate কৰি আছিল
glucose 6
Glucose
glucose 6
glucose
phosphate
6 phosphate okay
yes ma'am
are regulated internally some specific low molecular weight substances some specific low molecular weight substances such as product act as inhibitor so what you can add in the above reaction glucose 6 phosphate acts as an inhibitor inhibitor separately beleg inhibitor nahe yate reaction tute eta inhibitor form hoy jay mane product to inhibitor hoy jay okay where hexokinase is the enzyme yare agot ami ki dekhisu je ki hoy bahiror pora eta inhibitor ahe but yate ki hoy reaction tor endote jitu product hoy product to inhibitor hoy jay where hexokinase is an enzyme and glucose is the substrate. Is the substrate. বুজি পাইছা ডিফারেন্স টু ইয়াতে ৰিঅ্যাকশন টুতে কি হ'ব ইনহিবিটৰ আহি যাব ৰিঅ্যাকশন টুৰ যেটো প্ৰডাক্ট হয় হেটো ইনহিবিটৰৰ কাম কৰিব অলপ আগতে যেটো কম্পিটিটিভ আৰু নন কম্পিটিটিভ ইনহিবিটৰ আমি এক্সটাৰ্নালি এটা কম্পিটিটৰ আহি যায় অৰ ইনহিবিটৰ আহি যায় In the above reaction, glucose 6-phosphate acts as an inhibitor where hexokinase is the enzyme and glucose is the substrate. So we have covered the major part of enzymes. And next class we will discuss the questions of enzymes from your question bank. Okay, because enzymes part is very important. So I discuss for him. Before that, I our NCERT book. We'll go through the NCERT book of enzyme part. Clear everyone? The 3 types of inhibitor?
Yes, ma'am.
Okay.
Ma'am,
So let's
A2
come to
reversibly hobo na?
বুজি পানে কি কৈছা?
Ma'am, A2 reversibly hobo?
Yeah, eto reversible hobo. Okay, যেতিয়া negative inhibition to যেতিয়া আকৌ optimum মানে condition আহি যায় তেতিয়া কি হৈ যায় reverse হৈ যায়.
Okay, ma'am. Thank you, ma'am.
Acha, come to your NCERT book. Let us do one thing. Next class, we will discuss the NCERT book and we will discuss the questions from the question bank from the enzyme part. বুজি পাইছা? মই কি কৈছো NCERT book to read কৰিম next class ত আৰু enzyme আৰু just এটা point কব আছে হেতু কম plus আমি question bank enzyme part question discuss কৰিম মানে next Friday's class. Okay, and now we will go for the assignment discussion of animal kingdom.